Welcome, everyone. I'm Sandy Williams, president of Gladstone. I moved to remark that right now there are two places that have the really hottest seats in the Bay Area. One is across the bay at Oracle Arena, and the other is here, okay? So you're here to have courtside seats at some of the most exciting developments in all of science that's meaningful for medicine. I see many familiar faces and friends. Thank you for being here, and I see some new faces too. So I'd like to meet as many of you who are here for the first time when we have the reception after the event tonight. We've chosen a topic that most likely you've heard of and have some familiarity with for our theme for the symposium this spring. It's called Precision Medicine. I, along with many of my colleagues for decades, have dreamed of a medicine, a way of doing medicine in the future that moves beyond what sadly still today is the mistaken notion of one size fits all. We as physicians know when we prescribe a medication for someone that it's not going to work in everyone. But at present, we have only a few ways to know to tailor the therapy to the patient. I think there's some interesting movement to make medicine more precise, more personal, more preventive, more prospective, and we want to accelerate that trend. If you're aware of precision medicine, I suspect many of you know it through what it's meant for cancer therapeutics. This has been a really important area for development in medicine. We now can analyze the genome sequences of cancers or the patterns of gene activation and have some reasonable chance of targeting an effective therapy to one patient versus another. That process is still in its infancy, but it's growing. And I think if you know about that, that's good. You're on the dance floor to, I think, envisage the future of precision medicine. But what I hope you'll discern from the conversation we're about to have is that that's just the start. The notion of sequencing a single gene and having that predict what to do for a patient is going to work some of the time, but it's really just the start. I'm moved to remember when I first got into molecular biology some decades ago, the common mantra of how one succeeded in that business was one man, one gene, one lifetime. Some of you may remember those times. That was sad for two reasons. One, it took a lifetime to understand one gene, and two, it was mostly all men. So the one man, one gene was too literally true. Fortunately, we're changing on both of those fronts. We now analyze all 25,000 genes at once and all the hundreds of thousands of protein products that they produce, but it's not studying them one at a time that's going to really bring precision medicine to reality. It's going to be studying how they function in networks and revealing complex network interactions among genes, among proteins, and among what happens to us that affect our health and disease. And we have an all-star lineup for you tonight to give you some flavor of this emerging new world. Gladstone is doing its part in this world by a variety of approaches. We do it through our disease-focused institutes in heart, brain, and virology and immunology, but we're also doing it through a new program we call the Convergence Zone. The Convergence Zone is designed to create and bring to bear emerging technologies that allow us to ask questions about biology and disease in ways we couldn't do that before. And in so doing, to empower the disease-related work of all of our institutes. So tonight from Gladstone, we're featuring two of our scientists from our Convergence Zone, Katherine Pollard and Nevan Krogan. And I really have to hand it to Katie for having assembled the interesting conversation you're about to hear. We also have tonight invited, as we've done before, two leaders of emerging biotechnology and technology companies in the Bay Area. We, as you know, and many of you participate with us in doing this, believe that we'll only solve dread diseases for which we currently have no answers through fundamental science that unravels disease mechanisms, and then connecting that to biotechnology, pharmaceutical companies, device companies, informatics companies that can bring that knowledge to bear for the benefit of patients. So we like to engage with our partners from the industry side and hear the perspectives of how things are working. So again, I want to welcome you all here, and I'm going to ask Katie Pollard to get this started. And Katie, I'll let you, I'm sorry, it's Nevan Krogan to get this started. And I'll ask each of you to introduce our industry partners as you finish, okay? And then after they each give an introductory comment, we're going to gather on stage for some questions and answers to and from you.
All right, thank you, Sandy, for that introduction. As Sandy alluded to, I am a senior investigator in the Convergence Zone, which is led by Katie Pollard here at the J. David Gladstone Institutes, and my research is involved in developing and using tools which allow for the systematic, unbiased quantitative collection of biological information of various kinds. Most notably, we're looking at the functional and physical connections between different genes and proteins. And I think, as we all realize, we're in the middle of a genomic revolution. Pretty soon, we'll be going to the doctor's office and getting our entire genome sequenced. And as you know, there are large projects focused on different disease areas, different cohorts of patients, sequencing them or their tumors, trying to look for commonalities, sets of mutations that you can then link back to a given disease. But I think there's a realization now there is a limit in what you can extract simply from genomic data. And I think there's a strong argument to be made that we need to focus now on the next step, not the genes, but the proteins, as Sandy said, that are coded by the genes. And proteins often reside with other proteins in protein complexes. And I like to think of these protein complexes as the molecular machines of the cell. And in any given cell within us, a healthy cell, there are hundreds or thousands of these different machines that are functional. What happens in a disease state are that mutations arise that are introduced into these machines that make them not function properly. Hence, you get progression of disease state. Or you can think of it with respect to pathogenic organisms. Say HIV comes in and injects its proteins in your cells. And these viral proteins then come and hijack and rewire these complexes, these machines, in a way that's beneficial for the pathogen and detrimental for the host. So really the idea here is to generate this network of these machines, which I would argue strongly is a very powerful way to interpret this tsunami of genomic information that's coming online. And one of the ways to analyze these proteins and these complexes, these molecular machines, is through a technology called mass spectrometry. And here at the Gladstone, we have a number of these mass spectrometers which can monitor these molecular machines. This is a facility that's been sponsored by Thermo Fisher Scientific, the company in Santa Clara in Northern California, which arguably makes the best mass spectrometers in the world, and they've sponsored this facility. It's the second such facility they've sponsored, the first on the West Coast, and the first focused exclusively on studying different disease areas. And we're using this technology to study a number of different disease areas: cardiovascular disease, cancer, neurodegenerative disease, psychiatric disorders in different pathogenic states. And I would argue that studying these networks of molecular machines is going to be so crucial to make sense of the genomic data and ultimately let us come to precision medicine. I would like to go through a couple of vignettes of how we're trying to do this. And as many of you know, there's a number of large cancer genomic sequencing projects focused on different cancers. And what has been found here is that people with similar cancers or the same cancers, when you sequence their tumors and you look at the mutations, there's maybe 3 or 4 or 5 genes that are common amongst all of the patients. But the vast majority of these mutations are not similar. It's this long tail of cancer genomic mutations. So it's very hard to group these individuals together for prognosis purposes or to apply therapies. And in collaboration with Troy Adeker, who's the chief of genetics at the University of California in San Diego, we've looked at a certain cohort of patients that had ovarian cancer. We looked at the genomic data derived from the tumors. And again, looking at the genomic signature, they're very unique. But when you overlay that information onto these networks of machines, you can actually start making sense of this genomic data, and you can actually look for distinct categories. And we actually found 3 distinct categories by doing this. And these categories are very predictive with respect to prognosis, as well as which therapies would be responsive in a given group. So to me, that's getting at precision medicine. And we're doing this also with other cancers as well, looking at it in terms of the network of these molecular machines. A second example here has to do with HIV, and we've been collaborating with Steve Walensky at Northwestern University in Chicago for some time. He's been following a cohort of individuals that are essentially resistant to HIV. There's several hundred of them. And they're constantly exposed to the virus, but they do not get infected, which makes them very, very interesting. So what we've been doing is generating these networks of protein complexes that involve the HIV proteins, and we've identified a number of molecular machines which we think could be mutated in these individuals. And sure enough, when they looked in these individuals at their genome, they found mutations in these particular machines. And then we can go and study these in more detail, biochemically, biophysically, and more structural fashion. And hopefully then this type of analysis would lead to novel therapies, developing a drug that could mimic this mutation. This is a positive mutation because it allows you to be resistant to HIV, or ultimately using genetic engineering approaches, such as gene therapy or using CRISPR, ultimately to manipulate cells, introduce them into people, and then hopefully make them resistant to HIV. And I'd argue that these are 2 good examples of how you can go back and forth between the genomic data and the network data in order to get at the underlying biology behind different disease states, which would hopefully then lead to a better understanding and ultimately novel therapies. And the last thing I wanted to say is that this technology, and more importantly, this data, doesn't just connect different genes and proteins, it connects actually different people that are often siloed, focused on their particular disease area. So I would argue studying HIV is going to inform cancer, and vice versa. Because when we take a few steps back and look at the big picture, we're seeing these commonalities, these same proteins, say, being hijacked by many different pathogens or mutated in different disease states. So for example, we find a similar set of proteins in the host that are being hijacked by HIV and tuberculosis. So that would argue we should focus on those for novel therapies. But even as we go across from pathogenic states to other disease states, for example, there's a good correlation between the proteins being hijacked by HIV, the virus, and those same genes or proteins being mutated in different cancers, in particular leukemia and breast cancer. And to me, that makes sense. These are the Achilles heels of the cell. They can be mutated to result in disease, or they can be hijacked by particular pathogenic proteins. And I strongly believe, these type of approaches, again, will connect not just genes and proteins, but different individuals working on a different disease state, essentially breaking down the silos. And to accomplish this, I would strongly argue we need these type of maps, these reference maps, which will ultimately help make sense of the genomic data, which will get us closer to precision medicine. And with that, I think this is a good segue for Leah, because she's been focusing on a set of proteins, the chaperone proteins, which are actually linked through a number of different disease areas, and trying to develop drugs which could potentially have an effect in many of these areas, and ultimately trying to find a magic bullet or a silver bullet, if you will, to positively affect different compounds. So I'll let her come up to the podium and tell you about her very exciting work.
Thank you so much, Katie, for inviting me to come and speak. My name is Leah Makley, and I am one of the technical founders of a very small, nascent biotech startup here in the Bay Area called ViewPoint Therapeutics. We spun out of UCSF in 2014, and the company was founded based on some work that I did in my graduate research with Jason Gestwicki, who's now a professor in the Institute for Neurodegenerative Diseases just across the way. And so what I'd like to tell you about is how we came to work on the particular target. that we've chosen to address inside ViewPoint. So that target is called alpha-crystalline. It's actually, like Nevan said, what's called a molecular chaperone, meaning that it's at the center of a node of protein-protein, or it's the node at the center of a network of protein-protein interactions, and it's responsible for maintaining protein folding in a number of different cell types. So it actually is one of these very interesting targets that was identified across different indications with precision medicine. And it's been implicated very strongly genetically in cataracts and neurodegenerative diseases, in cancers, and in cardiomyopathies as well. And so basically what we decided to do was start to look at different mutations in this particular protein that are associated with these different disease phenotypes and build a biophysical screening strategy for finding small molecules that could actually bind to this protein and rescue these different mutations. So it was really building on precision medicine with a dearth of understanding of much about the structure and the function of this protein. What I think is really interesting about this particular system is that's oftentimes the case with targets that are identified this way, where we might not know much about what the protein does. It's probably not been drugged before. It may not resemble many of the typical drug targets that are oftentimes the focus of drug discovery campaigns. But we know from genetics that this protein is broken and that results in disease, sometimes in 100% of people who have that mutation. So there are a whole host of different proteins that have actually been identified in the post-genomic era due to precision medicine advances, where we need different sets of screening strategies to identify molecules that can actually correct those proteins. So alpha-crystalline is one of these examples, and that was why I was so intellectually interested in going after that particular target. So what we basically did was first, I guess I should back up and give you a little bit of background into the specific indication where we're working, which is cataracts. So the lens of the eye, for those of you who aren't very familiar, and I certainly wasn't when we started this work, the lens is this really interesting protein folding system where it actually consists of very, very high concentrations of soluble structural proteins called crystallins that are synthesized at probably the highest concentration of any protein in the body, 450 mg per mil or so, which is incredibly high, as those of you familiar with protein chemistry know. So what's also fascinating is that those proteins are synthesized early in development as the lens grows and matures, and then protein turnover stops. Those cells actually lose their organelles, they stop growing and dividing, and they lose the capacity to degrade molecules of protein and synthesize new ones. So there's an exquisite requirement for perfect protein quality control in the system and no metabolism with which to support that. So these proteins have to stay perfectly folded in order for the lens to be transparent. And as they start to fall apart and aggregate in defined ways, that gives rise to opacities in the lens, which are clinically diagnosed as cataracts. So we devised a screening strategy to find molecules that could stabilize those proteins and maintain their proper fold. And we're developing those as pharmacological treatments for cataract. So it's the leading cause of blindness in the world and currently only treatable by surgery. And we think we can actually use this technology to go in and intervene without need for surgery. Okay. What else? Yeah, the last point I guess I wanted to make is that this particular protein is a chaperone protein, meaning that also through genetic methods, it was identified that this particular protein is the key target in this system in particular, where alpha-crystalline actually binds and interacts with these other client proteins in the lens, and so by actually restoring its functionality, we stabilize the whole system. And so that understanding also came about through genetic methods.
Good evening. You've just heard from Nevan and Leah about protein networks and their importance in human biology and how their teams are figuring out how these are disrupted. The interactions in these networks are disrupted in disease and can potentially be targeted through therapeutics. I take it, at least for some of you, that was pretty complex. So I have some bad news. Unfortunately, what you're about to hear from me and from Peter is going to take that complexity and increase it by a couple orders of magnitude. So hold on to your seats and wish me luck in explaining this exciting topic to you. The good news is that the story that you're going to hear solves a variety of what were unsolved mysteries in human health and opens up this whole new universe of therapeutic options that we didn't have before. This is the story of the human microbiome. So let's start with a few facts. There are in each of our bodies trillions of cells. Trillions is a big number. That's approximately the same as saying there's one cell for every galaxy in the universe or one cell for every dollar in the national debt. So we're talking about a big number. And what you might or might not know is that only about half or maybe less than half of these cells carry your DNA in them. And the other half is a mixture of various kinds of cells from thousands of different types of microorganisms, including bacteria, archaea, fungi, and other eukaryotes, as well as parasites. And together, these make up something like 6 pounds of your body weight, and as I mentioned, could be more than half of the cells in our body. A lot of those cells in this complex mixture is in your gut, but they've been found in many other organs, including organs and parts of our body that we previously thought were sterile. So this really changes how we think about the human body and about health and our normal biology. And it really makes us think differently about precision medicine. We've been talking about looking at genetics and using that to understand why some people respond to drugs differently or different people get sick. Now, if each of us is carrying different microbes and each of those microbes have different genes in them, that means the vast majority of the proteins in your body that could be forming these machines that Nevan told you about are actually encoded in microbial DNA, not in human DNA. And that means that they're probably sitting in the cell of an organism that most of us have never heard of before. So I'm guessing everyone in the room has heard of HIV, influenza, E. coli, but raise your hand if you know about Bacteroides vulgaris. Anybody? All right, a couple people in the front row. You should. All of us essentially have this microorganism in us. It's highly abundant. It makes up a lot of the cells in your body, and yet you've never heard that name before, right? It's not a pathogen. It's one of these lesser known, what we call commensal microbes that's living in the human body, integrated with human cells, and not just sitting alongside of them, but really working with your human cells to make your body chug. So we didn't know very much about vulgaris or about the human microbiome in general a few years ago, but I bet most of you have heard, raise your hand if you've heard the word microbiome before, right? It's in the news almost every week, every day these days. So why are we suddenly hearing about it and yet our knowledge about what it is pretty limited? Well, this is a brand new field that's just opening up, and it's opening up because of two new technologies. One of them is next-generation sequencing, which enables high-throughput, low-cost genetic footprinting of the microbes that are in our bodies without the need to actually even know who they are or what they're doing, or to be able to isolate them out and study them in the lab. We can study them in situ in your body by looking at their genetic material. And this is really helpful because many of the microbes in our bodies can't be grown or studied in the lab by traditional microbiological techniques. But this is challenging because these genetic footprints are incredibly big and complicated data sets. If I were to get you to spit in a test tube and give me a sample and I wanted to figure out what was going on with the microbial proteins in your mouth, I would generate enough data to fill up about 10 laptops of computers or lots of cell phones worth of data. And this data would have many errors in it and biases. So it's much bigger and much more complicated than, say, studying the human genome. And one of the biggest challenges is that the microbes that are living on you are a community of different types. And so the DNA sequences come from different bugs, and we don't know which one came from which bug. When we get the sequences out, it's just DNA. And so some of that DNA is your DNA, and some of it's from Bacteroides vulgaris, but some of it's from some other bacteria, and I don't know which piece comes from which organism. And I don't get the whole genome. I just get a little 100 or 200 letters out of that genome. But I get 50 million or so of these little pieces, and I've got to put them together and figure out who's there and what they were doing. So this brings me to the second really important technology, which is high-performance computing. And this research would absolutely not be possible if it weren't for the advances in computing that allow us to implement algorithms that can mine through these massive data sets. My lab specializes in exactly this. We develop the bioinformatics tools and the statistical models for making sense out of this complicated microbiome data. And sometimes I feel a little bit more like a plumber than like a scientist, because I'm spending a lot of time figuring out how to get these huge data sets from one place to another through these clogged up pipes, which are the networks in this building and across the world that are connecting all of this data. So the IT infrastructure is really important too, in addition to the fast computers for being able to implement the algorithms. The data sets are so big, it's hard for us to even get them all in the same place to do a calculation on them. So this is a lot of hard work, but it's paying off. We've learned a lot, as you probably know if you've been reading these news articles on a weekly basis. The microbiome's involved in harvesting energy and vitamins and nutrients out of our food, in metabolizing drugs that we take. So when you take a drug, the amount that's actually available to your body will depend on what microbes you have. And we learn on a daily basis about a new disease or other trait of a person that's associated not with a genetic component of your human DNA, but with the genetics of your microbiome. Back to proteins for a little bit. So these microbial proteins are very important, and they aren't just sitting inside the microbial cells. The microbes in our bodies can secrete these proteins and in fact can inject them into the human cells so that they can form these molecular machines with human proteins. And so the exact networks that you heard Nevan talking about, now you can imagine instead of just human proteins interacting, or maybe 10 or 20 HIV or other viral proteins tapping into or hijacking, as he said, these protein networks. Now imagine that in this network, the human proteins are the smaller amount of the nodes, and there's all these microbial proteins that are connecting in and controlling that network. It starts to feel a little bit like the Matrix or something, right? The confusing and really challenging thing is that unlike your human DNA, the microbiome doesn't stay the same. It's different between people, and it's different in you on almost a daily basis. And so it's fluctuating so much that this network isn't even static. It's this moving target. So I warned you, right? This is getting really messy. But let's look at a few very specific examples where we can see that a biological process involves both a human gene or protein and a microbial protein. One is the connection between red meat and cardiovascular disease. Most of us know that it's not a good idea for our cardiovascular disease risk to eat a lot of red meat, but you may or may not know that we didn't really understand the mechanism through which red meat would increase risk for disease until very recently. And the reason was that the pathway that was linking things in the meat to the process that was creating the cardiovascular disease includes a step where it has to go through bacteria to create something called L-carnitine. And that small molecule is what leads eventually to the cardiovascular disease risk. So to be at a high risk for disease, you have to both eat a lot of red meat, but you also have to happen to have the bacteria that would be producing this L-carnitine. And that's been shown both in human cells and animal models, and then eventually in clinical studies in humans. Another example, touching on a different one of the disease areas that we work on at Gladstone, is about the immune system. So the microbes that live in our body, as I mentioned, are processing our diet. And one of the things they do is take fiber and eventually convert it through a molecular process into something called a short-chain fatty acid. And these short-chain fatty acids play a number of roles, including being involved in body odor, for example. But the important thing for your immune system is that some of these short-chain fatty acids are anti-inflammatory. And the microbes are making them on purpose. It helps your body not have an immune response against them. It's saying, hey, chill out the immune system. And in fact, it's necessary to have these microbes there and this high fiber in your diet in order to have a normal development of your immune system. And when the wrong kind of microbes are there, or when you don't have enough fiber in your diet, that process breaks down. The microbes start eating mucus that lines your gut lining, and that leads to the production of inflammatory compounds and inflammatory response. Finally, one of the more amazing connections that from my perspective, is that the microbes in your gut are also communicating with your brain through the vagus nerve. And this is two-way communication, that your brain is controlling what's going on with those communities, and those communities are also signaling back to your brain. And therefore, the microbiome has this new sort of emerging potential role also in neurological disease, as well as just psychiatry, stress, personality even, perhaps. So why is all of this relevant to precision medicine? The genetic differences between people's microbiomes are totally missed if you go in for a human genetic test. And however, they can be really important in terms of how to treat you and how you would respond to different drugs. And so the last thing I want to share with you is some exciting unpublished work that'll be coming out soon from my lab that's revealing a huge, even more than what I've already told you just now, variability between people in their microbiomes. So what the field has been doing up until now is footprinting the microbiome using genetic markers that it turns out are about this good. They would look at one of you, the resolution we have is to look at one of you and say it's a human or maybe a Neanderthal or maybe a gorilla. And so that's about how well we can identify these microbes. And that doesn't sound too great, right? I hypothesized that this would be insufficient, and in fact, it's really insufficient because microbes evolve much more rapidly than we do. So the difference, as a sort of gorilla versus human difference, is a huge difference for a microbe. It can mean that up to 50% or more of the genes are different, which is way more than the difference between us and a primate. And these aren't random genes, they're actually the transporters that are figuring out what's going on with the human protein network and bringing things into the microbial cells. They're the genes that are making these payloads, and then the genes that are taking them out of the cells, secreting them, or injecting them into your human cells. And so each of us differs in the way our microbiome is sending stuff into our human protein network. This can convert a microbe from being a pathogen, from being not a pathogen to being a pathogen. It can inactivate a drug or not inactivate a drug. And so this is really important. And so what the next step is, and what we're just starting to work on now, is to try to figure out what's driving these differences in different people's microbiomes. We know diet plays a role, your medical history, your lifestyle, but some of the effects aren't so immediate. They're over evolutionary timescales, or at least your whole lifetime. And we know this because when we look at people in different parts of the world, they have very different microbiomes. This is related, again, to your immune system. It turns out, if you think about it, most of us have a much cleaner body than our ancestors did. And when we look at people in other parts of the world with less access to medical care, they also have a more diverse microbiome. That's good on one hand, not so much infectious disease. On the other hand, our immune system was actually designed to handle all of that load. When we take it away, we turn against self, and there's a hypothesis that we and others are pursuing that that's a cause of the rise of autoimmune disease in industrialized countries. So this is important to me also personally because I suffer from 2 autoimmune diseases. So I'm motivated. I like the evolutionary story. I like the science, and I'm also very motivated. That's why I'm very excited to introduce Peter from Second Genome, the CEO and president, because his company is working on therapeutics that might be able to treat my disease.
Thank you, Katie. So I'm sure most of you are thinking, given all the complexity that Katie just described, what kind of idiot would start a company in the microbiome space and attempt to tame that complexity? So I'm Peter DiLaura, I'm that idiot. And thank you for the opportunity to be here to talk about the microbiome. It's a topic that I love talking about, and it's also, my wife claims it's the reason we don't get invited to many dinner parties. So I apologize if we go off track on these topics. So let me start, for those of you who study the classics at some point, you know of Narcissus, right? The hunter from Greek mythology who is in love with his own reflection. And at the risk of being slightly controversial, I think modern drug discovery has been narcissistic. Over the last 30 to 40 years, we've developed a tremendous ability to look deeply at host genomics, the mysteries encoded in our human DNA, and I think we've uncovered, as Sandy described earlier, just a wealth of information around the impact on human biology. But what microbiome science has catalyzed is an understanding that human health is not exclusively human. Those hundreds of trillions of bacteria that we're carrying around, the 4 to 5 pounds of mass in us that is microbial, is playing a really important part in our health and wellness, and it drives many important biological functions. And this is a community that has co-evolved with us since the dawn of humanity, but in the last, call it 70 years in our Western society, we've significantly perturbed this community, right? So we've changed the way we're colonized in early life. So kids delivered vaginally have different microbiomes than those delivered via C-section. We've changed our microbiomes through our diet. So if you have a Western high-fat diet, it's going to produce different microbiomes than those of a more traditional non-Western diet. And we've subjected our systems to tremendous amounts of therapeutics. So antibiotics obviously have a big impact on our bacterial community, but even non-antibiotic drugs have been shown to perturb our microbiomes in meaningful ways. So we now have the tools to start to understand some of these changes, which are really profound. And as Katie described, almost every week it's impossible to pick up a medical journal, and frankly, it's hard to pick up the New York Times without seeing some sort of article about the role of the microbiome in a disease. But much of this work that we have today is around correlative assessments. So the difference between disease and healthy and how that represents itself in different microbiomes, which is interesting. It's a good starting point, but in order to get to therapeutic interventions and in order to get to the precision medicine notion that we're discussing here, it's critical to get beyond those correlations to causal relationships and understand how mechanistically the microbiome and host are working together. So that's what, as a company, that's what we're focused on Second Genome. We've built a platform that allows us to look at clinical samples from patients across many diseases and understand what the microbiomes are producing in disease and wellness. What are the secreted factors, the metabolites, the proteins, the peptides that are coming out of the microbiome and then interacting with the host biology in a way that can either drive wellness or is exacerbating disease. So that's what we've done. Our first programs are in the clinic. Our first program is in ulcerative colitis, where we've identified a host target that is downstream of the bacterially secreted activities in patients with ulcerative colitis that we're inhibiting with that program. And we have the ability, within our own work, to extend our platform in multiple ways. But this work is only possible, as Katie described, because of the advances in sequencing technology, the advances in computational capabilities that allow us to start to make sense of these terabytes of data that we can now generate relatively easily. But the hurdle for us has now moved to the interpretation, right? So we're all living in an era where genomic data is rapidly decreasing in its cost. The accessibility of tools to generate these data is pervasive, but that interpretation step really is the next hurdle. So I think that in the context of the microbiome and thinking about how the microbiome matters for precision medicine, there are really three vectors. One vector is around a source of biomarkers, a way for us to subtype patient populations with much more resolution than one would get from exclusively genetic or genomic analysis of the host biology. The second area is around pharmacology. So there's a really interesting growing body of work that shows that the efficacy of drugs in some cases is dependent on the background microbiome of a patient. In one patient with one microbiome, a drug might be efficacious, but in another patient with a different microbiome, there might not be the efficacy of the same therapeutic agent. And then lastly, like our work, extracting the things from the microbiome that have therapeutic potential. So some people are approaching this in the context of the actual live biotherapeutics, right? The strains themselves being the therapeutics, or in the context of a fecal transplant, a whole replacement of a microbiome. We're instead focused on the secreted factors, the things that are coming out of the microbiome that can be developed as classic small and large molecule therapeutics. So that's our view of the microbiome, and I think tying it back to some of the conversation from Leah and Nevan and Katie, there's a huge tendency historically in drug discovery and science to be reductionist in our thinking and to drive toward very specific targets and to simplify our hypotheses in many ways so that we can conduct experiments that get us to endpoints. And I think that in many cases has served science and medicine quite well. However, we're at the advent of a space where that reductionist thinking, I think, is limiting, and we have to embrace some of this complexity and develop the tools and frameworks to deal with these incredibly complex multidimensional data sets, these incredibly robust systems biology frameworks. And so, those of us in industry on the biotech side are really dependent on the work that's happening at places like Gladstone, in Katie's lab, in Nevan's lab, to build us the frameworks to allow us to make sense of these data and to embrace the complexity that is inherent in our human biology, is inherent in what we represent. And so I think that this is really, for us, an interesting sort of point in our thinking about drug discovery and development to reflect on the complexity of this biology and start to apply it in very targeted ways therapeutically. So at the risk of not proving my wife correct, I'll stop talking about the microbiome, but I know that we've got an opportunity for some Q&A, so. Okay.
Thank all of the speakers. So I should have mentioned at the outset, we didn't do detailed introductions because there's a bio-sketch of all of our speakers and participants in your program, so I'd urge you to take a look at that. Are we having fun yet? I hope so. This is pretty exciting. I have to make a personal comment about Peter's reference to the dinner table. Some of you have heard my talk about inviting a mitochondrion to dinner, so this is a familiar topic to me. I thought I'd take just a minute and perhaps give you some conceptual hooks on which to hang some of the things you've heard about, and then we'll move to questions and answers. I think one of the ways I conceptualize what we are as human beings in a biological sense is, yes, we begin with our genome. We begin with our DNA. 3 billion nucleotides, A, C, T's, and G's lined up in every one of our cells. I thought that 3 billion number was big when I got to it, but it really is only the start, as you've heard. I think of life inside our cells as beginning with DNA. And remember, the central dogma of molecular biology, as Francis Crick characterized it, is DNA makes RNA makes protein. So yes, we are determined in part by our DNA, but that's only the start. The DNA actually doesn't do the work of the cell. The DNA gives rise to RNA, and each of the genes has like a rheostat on it that determines whether it makes RNA or not at a given time. So that next process, loosely defined, is gene regulation or epigenetics. So we have the layer of the gene sequence, the DNA, and then you move up in any given cell at any given time, and this is changing at all the times. You have the RNA that's made in that cell, and the control of that is epigenetics. And then you move up another notch into Nevan's realm, into the realm of the proteins. The RNAs make proteins, and these proteins, a given set of proteins is what tells a heart cell to be different from a brain cell or so forth, and it's also what tells a diseased cell to be different from a healthy cell. So at that level of the proteins is where the action really occurs. And the proteins that are there at any given time are important. How they are modified, because they get modified by other signaling processes. But the proteins do their work in these little nanomachines. So our cells are full of a whole variety of different protein clusters where proteins come together in teams to do this or that. And we've had some understanding that this is what happens for a long time, but only now are we beginning to discern this out in a near complete scale of everything, every gene, every RNA, every protein. So that's one step. So that's the beyond your DNA story. Now, all of that then makes up your cells. So remember, 3 billion nucleotides of DNA, we have somewhere about 50 trillion cells in our body. That's 5 more zeros on top of the 3 billion, right? So it's now a very big number, but that's matched by at least an equal number or perhaps even a greater multiple of these other things that live on and in us. So pretty complicated so far, right? This is why I always urge everybody to avoid being persuaded by simplistic notions of what you should eat or what you should take. It's much more complicated than what you'll read in newspaper ads. And then to just conclude by talking about the microbiome, I hope what you grasped is whatever you eat, goes into our intestine, and then chemicals from what we eat are absorbed into our bodies. And that's the way we understand nutrition, right? Well, it's not that simple. The chemicals that we eat are sometimes acted on by the microbiome before they go into our bodies, but lo and behold, not only that, when the chemicals come in our bodies and are acted on by our own cells, they sometimes go back out and are modified again, and a whole nother wave of signaling comes back. And those chemicals that are undergoing this process have profound effects. They can affect your mood, they can affect your memory, they can affect your propensity to have a heart attack by altering blood clotting. So what we're trying to give you tonight and have some fun with it, I think, is just a flavor of the complexity of what we have to deal with to try to solve diseases and make ourselves healthier, but a sense of hope that we're getting our arms around it, and these are some of the people who do it. So with that, I'll open the floor to questions. Yes, in the back, please.
Could you wait for a mic, please? Is there any relationship between antibodies and microbes?
What I heard is, the question, is there a relationship between antibodies and microbes?
Yes.
Okay, I think first we need an explanation of what each of those means, and then there is, in fact, a connection, but who would like to address that?
All right, so there we go. Great. So yes, there is a connection. So antibodies are made by your cells, and they are recognizing little pieces of proteins. For example, pieces of proteins made by microbes. They can also recognize pieces of protein made by your own cells, and that's where autoimmunity crosses over, as I mentioned earlier, and is related to the microbiome. The immune system can also recognize self in that way. It's a memory system for your cells for what it's seen before, and it helps your immune system to understand what's a pathogen and what's not, and how to deal with it.
Reg.
It's exciting that the microbiome affects so many things, but then you would expect that things like personality might be affected by broad-spectrum antibiotics. Has there been reports of that type of thing?
Sure, yeah. Let's see, clinical evidence that antibiotics affect personality and mood, I think, is probably pretty weak right now, but the anecdotal evidence is on the gut-brain axis and the role of the microbiome is growing. So I think when you think about indication spaces where microbiome science is most robust, you can probably start on one end with C. diff-associated diarrhea and the acute infection scenario there. And then you move to places like GI inflammation, Crohn's and colitis, metabolic disease. We're starting to see some good evidence in areas like obesity, maybe downstream liver like NASH or NAFLD. And then the gut-brain axis is, I think, emerging as another area. It's challenged because that's a space where the animal models are tough to interpret and extrapolate from. But you certainly see, I think, in animal models, greater anxiety measurements with antibiotic-treated animals and things like that.
Given the national epidemic in and obesity in children. Could you comment on the use of antibiotics in early childhood?
Yeah, well, so I'm not a doctor, nor do I play one on TV, but I think that I think that it's there's some data that suggests that early life antibiotic usage is in fact correlated with later in life obesity and metabolic disease. You certainly see it in animal models, right? So where antibiotic use can drive an obesity phenotype in animal models. So I think that it's something to consider, and I think it's both the clinical use of antibiotics, which I have young children. We use antibiotics when we need to because they've dramatically improved life expectancy across Western civilization. Right, they're good drugs for many reasons, but it's both clinical use as well as use in subtherapeutic doses in our food supply that's also relevant, right? So antibiotics are often used for in cattle and in poultry, and so I think it's been demonstrated in some studies that a therapeutic a dose of antibiotics that's equivalent to what comes across in our food supply in animal models can also drive metabolic disease phenotypes in those models. Katie, do you want to add?
Yeah, I wanted to add a couple other comments on obesity. So one is that we're involved in a weight loss study. So people who are already obese trying to lose weight and looking at the microbiome in that process, and how someone responds to a diet definitely depends on their microbiome. And we're doing this with collaborators who are studying people who are in an inpatient situation in a hospital where we can really control what's actually going into their bodies. And so I think there'll be some interesting stories there. On a less optimistic note, my lab, one of the more controversial or newsworthy things that we've done in the microbiome space was actually to debunk some of the associations between obesity and the microbiome. Not to say that there aren't connections, because you can take an obese person's microbiome, put it into a mouse, and the mouse will gain weight and change its metabolic profile. So there are connections, but when we look across populations at people, there is not a microbiome signature for obesity. So I can't look at somebody and measure their microbiome and predict that they would be obese or not. There's absolutely no signal. If you make a scatter plot, there's just a body mass index against anything that you can measure on the microbiome, and we've done this on thousands of different things you can measure. You just see like a scatter shot. There isn't a clear association across people. And that's probably because all of us have different diets and different human genetics and other things that are interacting with the microbiome. So this is back to Sandy's point about not expecting a magic bullet or an overly simplistic answer, which we would all hope for. Unfortunately, I think that's not the case. There was some early evidence that maybe there was a microbe that was causing obesity, but that's not the case.
Yes.
Sorry, I'm torn between two questions, but okay, I'll ask the serious one, and I'll ask the other one at the break. So we've had previous technologies that have come around that have led to an exuberance about how great it is to have all this data, and boy, we don't even need hypotheses anymore. The data will tell us. I mean, Affymatrix arrays, probably before that, it was, I don't know, maybe 2D Western blots. Is anything different this time, or are we still gonna have a lot of people doing dumb experiments on big data?
I think everybody should answer that one. You could.
Yeah.
That was a serious question.
So I can
It's a
comment on
serious
that.
question.
Where to start on that? So I would argue recently there's been some breakthroughs in terms of the quality of data that can be collected, in terms of how quantitative it is. So in the past, a lot of the information was qualitative, and we've developed technologies which can allow for very powerfully quantitative collection of a wide variety of information. So I would say there's a huge leap there in the quality of information that can be made. But to me, there's two major bottlenecks, and you're pointing them out. One is integration of orthogonal types of information. Okay, and Katie's one of the leading experts in the world on doing that, combining the proteomic, the genomic, the metabolomic type of information in a meaningful way. And related to that, I think even a bigger bottleneck is actually the visualization of this data. is putting the data in a format where people who have studied a given gene, the one gene, one man who's studied a given gene for 40 years. Not to say you're studied a gene for 40 years, Sandy, but
Almost.
So for someone like Sandy to look at the data in a way that they can generate hypotheses about the function of their given gene. And to me, that's even the bigger bottleneck, is how to put the data in a format that's actually digestible for the scientists to look at, to say, okay, what are the next steps? What do we have to do next to help understand the function of this gene or the underlying biology behind this disease state?
I have to make a comment here in reference to Nevan. For some time, I've gone to lots of talks, Chris, and I'm sure you're referring to some of this too, where some self-identified systems biologist comes in and shows you these vastly enormous data sets, and they show that, yes, you can measure that this is up and this is down, and you sit there through that and you say, that's dazzling, but you have to ask, where's the meat, right? What does it mean? One of the reasons I've gravitated towards Nevan Krogan is he's a systems biologist who delivers the goods. When he sifts through these enormous data sets of proteome interactions, there are pearls of biological insight at the end of that that are meaningful for how we fundamentally understand how important processes work or how we might overcome diseases. So we're very cognizant of the phenomenon you're describing and trying to not fall into that trap. Yes, Will.
So with the advances and the quantity and the complexity, how does Gladstone prioritize its objectives?
Okay. Well, I'd say we have two approaches to that. The first approach, the main approach, is to gather really clever people here and then not get too much in their way. That I really do believe is the fundamental basis for where most of the real advances in science will come. You cluster people, you give them a supportive environment where they have colleagues who challenge them, they can bounce their ideas, but don't try to over-engineer them. That's the first step, and that's, I think, part of the environment that was created here by Biomedical years ago, and that I think is a core part of who we are. However, there is a second part. When out of that, what I call a kind of random walk, curiosity-driven ideation, when out of that, insights emerge that we can look at and say, yes, that one can be put on a path to fix a disease, potentially. It's a long way from there, but there's promise there. Then we have a different approach, and then we cluster and form teams. Then we find industry partners, and we try to drive that knowledge out to be something useful. So I think we prioritize first on having great scientists, and second on having programs to move discoveries out towards an advance in medicine rapidly.
I have a question. So is it possible to completely modify someone's microbiome by doing a microbiome transplant? And how common are these therapies over the traditional way of using drugs for treatment?
Great question. So there are efforts to curate or manipulate or replace someone's microbiome. It is very difficult to eliminate everything that's there, even by taking a broad spectrum antibiotic. And so it's not so much that you just cleanly replace something. It's a much more nuanced process. One of the ways that's actually being used in the hospital here in UCSF and that one can get a treatment is to have a fecal transplant. So you get the whole microbiome of someone else who is healthy put into a sick person, and that has been able to cure certain diseases. As Peter mentioned, it's been most successful for an acute infection by a type of bacteria called Clostridium difficile that's life-threatening. And then giving a microbiome transplant can, in many patients, cure that disease, to get the C. difficile under control. But it's not such an easy thing to replace someone's microbiome. And I think that's why, in large part, Peter's company is focused on more targeted manipulation and addition and subtraction of the things that the microbes make, rather than trying to manipulate the cells. Do you want to add to that?
Yeah, and I think there's a growing effort to use the fecal transplant approach in other indications, chronic indications, that's been met with mixed success. And I think that the data is building that suggests that it's important to not only be able to match sort of donor with recipient, but also to create a sustained effect over time based on the host biology that's driving the specific disease, right? In an acute infection, you've got C. diff, you've got it because you went into the hospital for knee surgery, you had high-dose antibiotics, it knocked down your existing microbiome, and then you got this hospital-acquired infection that was able to take advantage of the fact that it was working in an empty ecosystem. It was able to proliferate very quickly, and the wholesale microbiome transplant actually appears to be very efficacious in creating an ecology that the pathogen cannot survive, or is not symptomatic if it persists. But that's very different than chronic disease scenarios where your microbiome has already sort of tipped over the host biology at some point, and they're signaling back and forth between the disease, the host disease, and the microbiome, and so. Sort of a wholesale manipulation hasn't been shown to be broadly efficacious in those settings. It's not to say it couldn't work over time, but it appears to be a more sort of sophisticated precision alignment than in an acute infection scenario.
I'd like to get Leah into the act here in a moment, and I'd like to do it in the following way. Again, some of you may have heard me say this before, but in my efforts to bring non-scientists into the kind of visceral excitement about molecular biology that many of us feel, I like to speak to the aesthetics of understanding biology as well as to the practical applications. I liken enjoyment of a talk about complex biology as being somewhat akin to eating oysters. The first time a naive person looks at an oyster, you'd go, that's not for me, right? But when you acquire the taste and you pair it with a good Chablis, you know, what can be better? I want you all leaving the room tonight that you've learned to eat the oysters. And so here was the comment about Leah. To me, one of the beautiful things about biology are some of the surprises about how things work. So she works on this so-called chaperone system that involves alpha-crystalline. And she talked about how the lens of our eye, to be clear, has to have its proteins exactly arranged. Anything that's messed up and you're not going to be able to see. You got a blurry system. So we've evolved a system to quality control the way these proteins sit in space. Now, there's another important cell in the body that requires a similar perfect array of proteins lined up. Who can tell me what that is? Throughout almost the whole cell, a set of proteins have to be lined up for us to live. Okay, I'll give you a hint. Have you ever heard a talk by Deepak? Ever seen his pictures of heart cells? Yeah. So what happens in heart cells? You have these so-called sarcomeres, and when you label them with fluorescent proteins, they have these beautiful arrays. I mean, they're just gorgeous, but they've got to be perfect. They've got to be exactly aligned. And if nature comes apart and those proteins can't be there in just the right number and of the right type, then the heart doesn't work well. So what happens when alpha-B crystalline is mutated? You get a cataract and your heart doesn't work. I mean, who would ever thought that? And something similar takes place in brain cells and all the other cells too. It's just in the lens and in the heart, it just particularly hits you in the face, and the same system is required. But now, go on from that. That's the aesthetics. You're working on doing the useful part.
Yeah, so I'm glad that you drew the connection between the lens and the heart. So we're actually taking what we've learned about this particular protein structure and function and what's required for this array of very tightly packed protein molecules in the lens, and learning, basically taking what we've learned about how we can make sure that that stays intact in the lens, and now taking that knowledge and applying that to cardiomyopathy as well. And so I want to maybe spend a few more minutes talking about how exactly the system works, because I think it's really, really interesting.
Yeah, who would think it, working on cataracts and fix the heart? I mean, but this is how biology works.
So in the lens of the eye, right, I said that these proteins are actually synthesized early on in development, and then the cells in the lens lose their organelles. If you had organelles, if you had mitochondria and a nucleus and a ribosome inside those cells, they wouldn't be transparent. So it's evolved to actually be this tissue that's the only tissue in the body that has perfect transparency. And this arrangement of protein is required for that transparency. So it's like biological glass. It's a really fascinating system. Those proteins also, in their proper folded state, give the lens just the right refractive index to be able to focus light on the retina, and also give the lens its tensile properties that allows you to see near and far distance. So the lens actually changes its shape. But since those proteins last as long as we live, and the lens is relatively exposed to the environment, right? Those proteins actually undergo a lot of changes over decades that can accumulate to eventually destabilize the fold. And you see this a lot with lifespan extending, where these diseases of aging, we're kind of outliving our normal protective mechanisms, right? And so if you live long enough, you will get cataracts. Like if you live long enough, you're very likely to get certain other protein misfolding types of
So
diseases.
it's question time for me again,
Go ahead.
okay? You heard Leah say the cells of the lens have to last as long as we do. You know, they form when we're very young, and then you don't make any new ones. There's two other important systems where we don't make any, or at least not very many, new cells after we're newborns. Who knows what those two are? What does Gladstone work on in part? The heart? The heart cells, unless Deepak regenerates them, are pretty much the ones you have when you're a few months old. And Leonard Mookie is here, and you got the brain. So the brain, most of our neurons are cells that are formed early on in life, and they have to last a lifetime. And there, too, when the protein structures in the brain are at all amiss, that's the fundamental basis of much of neurodegenerative disease. So isn't it beautiful how it all connects? And yet, yeah.
We've got a question back here.
Yeah.
I'd like to follow up on Sandy's point. You paid a lot of attention to children that have early onset cataracts. Do they have other protein folding dysfunction? Do they have cardiomyopathies or anything of that sort?
Yes, absolutely. So we started actually working on this target because there's a mutation that causes hereditary cataracts in children that was identified in the 90s in several French families, actually. And those children actually are born with cataracts or will develop cataracts in the first few years of life. But the much more serious phenotype that goes along with those cataracts is actually cardiomyopathy. So this particular protein is ubiquitously expressed in many different tissues, and it happens to be most important in the lens and in the heart. So yes, and that's also true for many of the other mutations that are associated with hereditary cataracts.
Can I touch on something?
Oh, sure.
It's related to
Nevan.
these questions. It's actually related to what Sandy was talking about in terms of, studying cataracts informs studying heart disease. And I would argue right now we're at this this biological revolution, and technology is really driving it. And unbiased data is being generated across all these different disease areas. And I really believe the big discoveries are going to come from, as I alluded to during my talk, when groups that study, say, heart disease interact with groups that study cancer. There's going to be these commonalities that exist, and there's going to be treatments in disease X that could ultimately apply to disease Y and vice versa. And the technology and the data derived from the technology is going to allow for these connections. And just a comment about the convergence zone. I know the Gladstone split into these three institutes, but the convergence zone, the real goal of this is to bring all these technologies here to be cross-cutting across these different disease areas here at the Gladstone and beyond, And I'm really excited to be part of this new entity at the Gladstone, because I really believe that there's going to be some very big discoveries being born out of this technology in the very near future.
I see Warner Green here, who heads up our Virology and Immunology Institute. You heard Nevan speak at the start about how when viruses invade our bodies, they produce proteins that then hijack some of these machines. Nevan, you want to comment how that's opened the door to potentially new therapies, and how that might that approach might be used to protect us from emerging viral threats?
Sure. I am affiliated with Werner's Institute, and we've been targeting a number of different pathogens using this approach, looking at the molecular machines that are being hijacked by different viral proteins. And our entry point into this work was looking at HIV, and it was really motivated by the fact that a number of these HIV proteins could not be studied in an in vitro setting. You could not get a crystal structure of these HIV proteins. And if you had the crystal structure, you could hopefully develop more intelligent targeted drugs to, say, interrupt the function of that HIV protein or a particular interaction. So what we did was unbiasedly try to identify the human proteins that were being hijacked by these HIV proteins, which reveals a lot about the biology of the infection, but it also allowed us to identify these proteins that allowed us to reconstitute these complexes in an in vitro setting so that we could crystallize it and actually look at the complex and say, here's the HIV protein, here's a pocket that maybe a drug could be targeted against to interrupt the function of that given HIV factor. So that has been very revealing, trying to get more mechanistic, a deeper understanding behind the different HIV proteins. And as I said during my talk, we're starting to look across different pathogens now and trying to look for commonalities. And the idea is, okay, let's look at a protein that's being hijacked by tuberculosis, hepatitis C, and HIV. And we actually have an example of that. So can we develop a therapy that would have this kind of broad spectrum results going forward? So it's you know, as Sandy was alluding to before, you can use this information, go very deep mechanistically and get information, but then you can look across broad and look for these commonalities and also get information as well. And it's really going back and forth between these two viewpoints, which allows us to understand complex biological phenomenon behind different disease states.
Granted that there are fatal diseases, cancer and some microorganism infections and that sort of thing, but wouldn't the microbiome have evolved over time to depend on the survival of its host? And in that sense, isn't there a bound on this whole thing that why doesn't our microbiome fundamentally want to keep us alive as long as possible?
Yeah. So I think that it likely does, and if you take the multi-millennial, generational, evolutionary arc of our microbiomes, it has evolved with us in a commensal synergistic way. But the Western society perturbations of the last 50 years are really just a blip on that evolutionary scale. And I think the hypothesis, and Katie can certainly talk to this in more depth, I think the hypothesis is that those perturbations have been pretty rapid, and the microbiome evolutions have not been able to keep up effectively.
Yeah. So one thing I've been thinking about ever since I was an undergraduate is about how culture and things that we do as people can evolve so much more rapidly than biology, and even the biology of microbes. And they divide much more frequently than our cells, and some of them are super mutators. And we think of them like sort of immediately responding when we put a new antibiotic out on the market. Even microbes are evolving pretty slowly compared to cultural evolution, which is just going faster and faster. And so I completely agree that the system has the properties you described, and we are in a moment of disequilibrium right now, essentially.
One more question.
Was wondering if you guys are doing most of your work in petri dishes, or have you moved on to some live humans? If that's.
I work mostly with files on a computer, but I'll let the others comment.
Well, most of our work is done in cell lines, but we are now, as I alluded to, interacting with the clinical world, where there's these cohorts of individuals that are being monitored for many, many years in terms of being, resistant or susceptible one example for HIV. And can we marry the genomic data with this kind of the systems approaches that we take? And we have a similar project ongoing with influenza as well. There's a cohort, again, on the East Coast that we're trying to marry our systems approaches, our data that we derive from cell lines, trying to marry that with the clinical data that's coming from different cohorts. So I work with humans a little bit.
Yeah, I'd comment that at Gladstone, almost every day, somebody here is working on a problem at the level of single atoms, all the way up to molecules and cells and organs and model organisms, human materials, even out to populations and ecosystems. We're not doing any extraterrestrial life work here yet, but we pretty much cover the rest of the spectrum. Okay.
Sandy,
And all your files are human, most of them are.
Yeah.
Yeah, it's mostly human. The mouse. We are doing some work, and I know Peter's taken this approach too in his company. We're using mouse models when we want to look at a longitudinal process, because that can be a very slow evolution of a human's health breaking down, and we can study that much more rapidly and try to get at the cause and effect in a mouse model.
Yeah. Yeah, and our starting point is almost always human clinical samples. So we work with institutions and Mayo Clinic to get patient samples from disease cohorts and healthy patients. And then we go genomic analysis and to in silico analysis from there. But it's very important for us to then get back to the in vitro and wet lab work and ultimately into the clinic. So our lead program is in phase 1 human clinical studies right now through the single ascending dose piece of that. And that's obviously where the rubber meets the road on all of this stuff.
Okay, one more question.
thank you so much for this wonderful, wonderful evening. I'd like to know, you used the word hijack proteins. What happens with dementia? And of course, very, very interestingly, how much you're facing that most of us in this room will live to probably be 100.
I mean, there's experts here in the room that can speak a lot more appropriately about this than I can, but a lot of this is related to protein aggregation. And there's still some mechanisms that need to be worked out. What is the actual, the next step after protein aggregation? There's a number of theories here. But, I mean, I look at, you're talking about hijacking, I look at a mutated protein the same way, the protein from us, the same way I look at, say, an HIV protein. It comes in, it's going to do some damage, it's going to cause some trouble. And, can we effectively fight off the adverse effects from it, right? So conceptually, I think of mutated genes the same way as I think of viral proteins or pathogenic proteins, in that, as you say, the hijacking, the mutated gene can hijack the system and cause problems and ultimately result in disease states. So from an experimental point of view, I see these as the same, and I think that's a good thing in my mind.
I'll quickly get out of the way and not be the barrier to food and drink, but I did want to do a couple things. First, I want to thank our panelists, and I appreciate that. And secondly, I want to thank all of you for being here and participating in such an energetic manner. All of you are special, but I think there's some people who are quite special to us whose names are in the program, and some of them are here tonight too. I won't ask you to stand, but everyone should know we can't do what we do without the kind of community support we get from our foundation board, from our President's Council, and from the trustees of the Gladstone Trust. So let's give all of you and them an applause too. Okay, well, thank you all, and we have a reception.
In writing.